Hi everyone, I'm really excited to be talking about RAPPPID, which is our method for improving interaction prediction on unseen proteins. If you'd like to download the slides and references for this presentation, simply scan the QR code on the screen. My name is Szymborski, and I'm from McGill University's Department of Electrical and Computer Engineering. I'm a member of MILA, the Quebec AI Institute, and I'm a student in the AminoMads Combine Lab. So let's begin by talking a bit about protein-protein interactions. It can be useful to think of biological processes as an undirected graph of protein-protein interactions, or PPIs. It's an incomplete model, of course. There are plenty of other macromolecules and factors that make up these biological processes, but these assumptions have historically gotten us pretty far. So to think of PPIs as an undirected graph, simply think of proteins as nodes in the graph, and the interaction between proteins as edges between these nodes. These graphs can get rather complex rather quickly. Here are just some of the signaling pathways we understand to be involved in cancers as charted out by the KEGG dataset. So how do we identify these interactions? Typically we use so-called wet lab experiments, such as co-immunoprecipitation or yeast-2 hybrid experiments. These experiments usually take days or weeks to complete, require expensive reagents, and produce a lot of plastic waste. These experiments, however, give us a lot of confidence regarding the ability of two proteins to interact. Predicting protein interactions using computational models try to address some of the limitations of wet lab experiments. They take seconds or minutes to perform, and they are low to no cost. They do, however, consume electricity and produce e-waste, and they are not evidence of interactions. They are indeed predictions of proteins interacting. The setup for the protein-protein interaction prediction task is, given two proteins, do they interact? How well do existing methods do on predicting these edges? Well, it depends on how you measure it. Park and Marcotte in 2012 investigated the validation methods for PPI interaction methods of the time and came up with some interesting findings. Most methods in 2012, and indeed still today, use what Park and Marcotte called C1 validation sets, whereby edges in the graph are randomly chosen to be held out as part of the testing set, and another random sampling to be held as the training set. Methods tested by Park and Marcotte at the time scored quite well, and indeed these were in the range of their first reported scores. This is, of course, for C1. Park and Marcotte, however, devised what they called a C2 dataset, whereby nodes are randomly labeled as training and testing nodes. Testing edges were required to have at most one node that was also found in the training set. The methods tested on C1 were tested on C2, and had their AROC scores drop severely, as you can see. Park and Marcotte described a final and stricter C3 dataset, whereby testing edges were not allowed to have any nodes that were present during training. Methods tested on C3 fell even closer to random AROCs on this binary prediction. These results underscored the prevalence of data leakage in the way PPI prediction tasks are typically validated. The question then becomes, how do we construct models to address this problem? This is where we'd like to introduce RAPPPID, a method for the regularized automatic prediction of protein-protein interactions using deep learning, which was recently published in Oxford Bioinformatics. From the outset, the setup for RAPPPID's architecture isn't very different from many other deep learning methods for PPI prediction. We first begin with the amino acid sequence of proteins whose interaction we wish to predict. The amino acid sequence is then tokenized using the sentence piece tokenizer, which chunks the sequence into tokens of 1 to 4 amino acids. Sentence piece uses a probabilistic algorithm, and the tokens generated for a sequence is not deterministic. This helps to regularize our model at the token level. These tokens are then used as input for the averaged weight decayed long short-term memory encoder, or AWD LSTM. This is a standard LSTM encoder that employs a number of regularization techniques that are appropriate for LSTM models. RAPPPID is a twin architecture, which means the weights and architecture of the encoders are shared for both protein A and protein B. The output of the encoder is then used as a hidden representation of both protein A and protein B. These representations are then concatenated and passed to a fully connected classifier network, which outputs a float between 0 and 1, predicting interaction. Now, as I said, the overall architecture of RAPPPID is similar to many deep learning methods for PPI prediction, but there are many important differences in training that I've alluded to. Mostly RAPPPID employs a judicious use of regularization that is appropriate for the architecture. This includes AWD-LSTM, embedding dropout, and stochastic weight averaging, or SWA. It also uses sentence piece tokenizer, which I spoke about earlier. Now, you may be familiar with dropout, which zeroes out neural network activations with a random probability. Dropout, however, has been found to severely degrade the ability of LSTMs to retain memory. To address this, we use drop connect, which instead zeroes out weights in neural networks with some random probability. This has been shown to be a far more appropriate regularization for LSTM networks. We also use embedding dropout, which is a method whereby tokens are randomly zeroed out to help prevent models from becoming too fragile and placing too much importance on a single token. The A in AWD-LSTM refers to averaged optimization. Here we use stochastic weight averaging, or SWA. SWA is similar to averaged stochastic gradient descent, but keeps a pair of weights. One that the optimizer minimizes, and the other that is a running average of the previous weight. This helps the model to overcome, for example, rather flat minima. So how does RAPPID perform? Here we compare RAPPID to three other methods. Sprint is a sequence similarity method, which was designed for C2 and C3 datasets, and Deep PPI and Piper, two deep learning methods. We can see that in the C1 case, RAPPID performs comparably to leading methods on the string PPI dataset for Homo sapiens. In the C2 case, we see that RAPPID retains much of the performance relative to Sprint and Deep PPI. Finally, in the C3 dataset, RAPPID does a far better job than the other three methods at maintaining its performance, scoring an AUROC of 0.803. We performed an ablation study on RAPPID to determine which elements of RAPPID were most important to its performance. We found that the sentence piece tokenizer and the AWD-LSTM encoder to be the most important parts of RAPPID's performance. We also tested replacing the AWD-LSTM encoder with a transformer architecture. We tested two sets of hyperparameters, a larger transformer based on the BERT architecture and a smaller transformer. Both resulted in worse AUROCs, which we attribute to overfitting caused by the relatively small size of our datasets. We also tested RAPPID's amenability to transfer learning by pre-training the encoder on the string PPI dataset and fine-tuning the classifier on a protein-ligand interaction dataset derived from PDB named BioLip. Using transfer learning, RAPPID was capable of achieving an AUROC of 0.9089 on the BioLip dataset. We wanted to test how one might use RAPPID to validate a hypothesized interaction between some target protein and a candidate therapeutic protein or peptide. We used two examples, trastuzumab and pertuzumab. Both of these are humanized monoclonal antibodies that are used in the treatment of HER2-positive metastatic breast cancers. What you see here is a distribution of the interaction probabilities between trastuzumab and all the human proteins in the UniProt dataset. On the x-axis is the interaction probability, and on the y-axis is the absolute number of proteins with that probability. Here we see that HER2, which is our target, is within the first percentile of proteins predicted to interact with trastuzumab, with a predicted interaction probability of 98.8%. Similarly, for pertuzumab, our target is within the second percentile. Thanks so much for your time. I'd like to thank the members of the Combine Lab and our generous supporters.